got the right tools.
School for Transgenic Aesthetics Limited. I'm not kidding about the limited. I do think transgenic aesthetics are limited. Like, limited by what a body can do and what you can do to a body. We asked for a permit to make transgenic bacteria here, and we're waiting. Oron Katz is here, and we're going to sort of collab with you guys to get our hands wet and figure out what tissue culture is. Like most of these labs, the basic idea is to A, present you with the technology, B, present you with the dilemmas that the technology seems to bring up for most people, and C, let you think about that more than science. Not so bad. One strange excuse that we use is to say that you're not making science or facts right now, you're making a piece of art. What does that mean? What's the difference? Actually helps inform the debate. In other words, art is perceived of as being a frivolous thing that just sort of supplants society with some sort of new excuse for being, but not necessarily anything political, anything moving, anything, actually anything at all except for advanced design and headiness. Okay, this is a very sort of nihilistic view of art. I happen to have it sometimes I don't. Obviously, I'm referring to the libidinalization of biology. I think we're alive, and that's libidinal to me. And anything that's done within the field of life or living-ness is, to me, loaded with the erotic, one way or another. And when you get to tubes and things that inject and vents and various different sterile tools, to me it ends up being a certain kind of fetishism. I'll let you figure it out what it means to you, okay? So without further ado, I'm going to introduce Oron Katz to give a sort of brief history of tissue culture. Yeah?
My name is Oron Katz. I'm the director of SymbioticA at the University of Western Australia. But I started my art practice back in the mid-90s with a specific interest in tissue culture. So I got into this idea that tissue culture might be an interesting and an evocative way of engaging with life and open up different types of discourses in regard to the level of the manipulation of life and what it really means to us to do it now. But in a sense, and hopefully tonight we'll have an argument about it, I think that tissue culture is the most fundamental shift that happened in regard to our relationship to life, and that's what really ushered modern biology. And there's forgotten history that seemed to be driven somehow by the fact that there's much more interest in the last at least 30 or 40 years in another level of breaking down the body, that's a molecular level, although the cellular level and the level of the tissue is what really ushered it, as well as ushered this perception of the fact that the body is a raw material for us to engineer. And from that, anything that we see in modern biology and modern biotechnology really came from. So what I'll do, I'll give you like an historical narrative to make this point. And then we'll go straight into the hands-on work. So as you've seen also in the Wikipedia entry, the credit for tissue culture was given to Wilhelm Raux in 1885, who was able to, for the first time, sustain parts of animals alive outside of the original context of the body from which they taken. So it was alive for a short period of time. 1895, again, I've seen it in the Wikipedia entry. 1895, there was a realization that the body is becoming a raw material. So if before that the whole idea of natural history, the understanding of bodies was to do with observation, now we move into a realm where life is a raw material. Life can be something for us to engineer, so something that we can use for our own purposes. Again, tissue culture is really the idea of fragmenting bodies and maintaining those cells alive. it's not surprising in a sense, although here Harrison says it is surprising, but in those early stages of tissue culture, there was lots of media attention. It was a very interesting area for the popular media to look at those doctors, those surgeons, those scientists that were starting to take the body apart. Actually, the work was referred to then as artificial life, which is interesting. This term was forgotten for a while before it was reintroduced in the 1980s to do with computer-generated life. Because that's where it was, Coney Island, and a freak show. So the first premature baby ward in the United States, which lasted for 40 years, was a freak show in Coney Island, next to the bearded woman and all of the other oddities of life. And you can see it says there, infant incubators with living infants. Come and see them. And so this kind of this relationship between life and technological bodies also is there's lots of tension there, and it's, I suppose, what's interesting that not surprising that artists engaging with this connection tend to generate also quite a lot of freak-like excitement and interest in this type of work. They're putting quite a lot of different things together. But in that book, he also was referring to the fact that some undesirable elements in human society should be disposed of economically in gas chambers. So he was the first in the sense to recommend the use of gas chambers that was picked up by Germans shortly after. And we know what happened then. So after the war, the first successful cell line was developed. So up to now, scientists were removing cells directly from the organism and culturing them. Now most cells that are taken directly from the body have a finite amount of division they go through. It's called the half-life effect. So each time they divide, a short bit in the end of the chromosome falls off until there's nothing else to fall and the cells cannot divide anymore. But there are types of cells that can divide forever, and those are either stem cells or cancer cells. And so the first cell line, the first continuous line that actually started from an individual cell was the strain HeLa mouse that was isolated back in 1948. And those cells are still in circulation and are being used quite a lot for research, or at least as a tool. So if, just as a very rough estimate, if you would collect all of those cells from that individual mouse that died 60 years ago now, it will fill this room plus. There will be more cells now, filling this whole room out of this very one little cell or little mouse. In 1951, the same thing was done with humans. The first human cell line was isolated from cervical cancer of an African-American woman called Jeanette, and those cells are known as the HeLa cells. And we can talk a bit more about them because we are going to work with those cells today. So we are going to work with what can be considered a fragment of Henrietta Lacks that was taken from her back in 1951. We'll see how it makes us feel. 1954, tissue culture became much more standardized. So up to then, it was again a very messy operation. The scientists had to really, make their own liquids, to set up their own systems. And in the 50s, quite a lot through this book, The Cultivation of Animal and Plant Cells by Waag, this whole area became standardized. But at the very same time, he was extremely critical of Alexis Carrel and the way Carrel was doing his stuff. So you can read here that he sought to strip away the study from the study of this subject, the former atmosphere of mystery and complications, the grey walls, the backgrounds, the masks and hoods. So he's kind of going on about the decor or the mise-en-scène of this tissue culture and how problematic that was for the standardization of that field. Then, obviously, another major breakthrough in regard to moving fragments from one body to the next, the first successful kidney transplant in humans. 1978, another breakthrough, if you like, in this area, and that's the first successful IVF treatment, test tube baby. No genetics there, just tissue culture. Keep that in mind. 1991, the very same cells that we referred to before, the HeLa cells, was a realization that actually they represent something different. We don't, as biology started to grapple with the fact that there's no systematic way of looking at those fragments of bodies within modes of classification. Van Vellen came up with this idea that the HeLa cells, those fragments from Henrietta Lacks, should actually be referred to as a new life form, a new microbe, a new form of life that is originated from human, but is now a separate life form that lives in a particular environment. In this case, the environment is the laboratory environment. And as you see today, the HeLa cells are growing here. So, most of the galleries. 1995, the publication of the paper Tissue Engineering by Robert Langer and Joseph Acanti. And what happened there was the realization that cells can be more than just exercising futile freedom. They can actually be functional if we take them outside of the body and then we introduce them into the body. So this is really the birth of regenerative medicine. The idea that the body is a regenerative site and we can repair the body using the body-owned materials and cells. So the idea of regenerative medicine was playing on the notion that we are all closet salamandras. That salamandras have the ability to regenerate. If you cut almost any organ from the salamander, it will grow back. And we have those powers hidden within us, but we just have to find ways to unleash them. And so that was kind of the promise and the metaphor of regenerative medicine. But I would argue that we are really more like slime molds, that the moving and shifting of cells around is more like the way slime molds, which are very interesting organisms that live very happily as a unicellular organism, like an amoeba, and then once in a while they come together to create this complex multicellular structure, an organism with specialized areas. So we are like that. This whole idea of regenerative medicine is about shifting and moving cells around. It's not about regenerating limbs like the salamanders. So in a sense it's much less sexy, but that's what we are. Here is a model of how the body could have been repaired. That's from the 1980s, that's from 1989, where failing and missing and dysfunctional body parts would be replaced by mechanical apparatuses. So the heart would be replaced by a pump, and we can get bionic eyes and ears and legs and so forth. And then 10 years later, the idea was no, we actually use the body on cells to do so. So just the very basics of tissue engineering, this idea of functional three-dimensional tissue, the premise was very, very simple, and obviously it doesn't work, but it doesn't work for medicine, but works really beautifully for art. And that's this idea of producing a three-dimensional object out of those special biodegradable materials, those polymers, in the shape of the organ you're trying to replace. You then seed it with cells. This slide is actually from Joseph Acanti's presentation from 2000. So in a sense, that was before the stem cell hype. So he's talking here about specialized cells rather than stem cells that can be differentiated. And then you see those cells onto the three-dimensional scaffold outside of the body, and you grow it outside of the body for a while, and then you put it back into the body. So in a sense, bringing this dream of Alexis Carrel of repairing the body, almost like a mechanic, bringing this dream forth. Although again, I would stress that that was the promise started in the mid-90s. We are 15 years later, and we're not even close to achieving this type of complexity of repairing or replacing complex organs. But as a proposition, I think it's an evocative one and something for us to think about, because in particular, if we think about growing things which would never be reintroduced into the body, so growing functional tissues for other purposes. Anyway, we leave it there because we have a very busy afternoon today. We are going to work both with primary tissue, so cells taken directly from an organism, as you can see here, which would have a finite amount of divisions, I would imagine, if we have any divisions out of them. And we work with the HeLa cells that, as Adam mentioned, he received this morning from Leiden. So it was an early trip for him, and he got some cells which are still frozen in dry ice, which is another really interesting thing about tissue culture, because tissue culture breaks down quite a lot of conventional barriers that we perceive in regard to life. So first of all, we would be able, in theory, to grow the HeLa cells and those cells together. You can mix and match cells from different organisms. There's no immune system, so you can basically co-culture almost any type of cell with any other type of cell from different origins. most tissue culture exercises are using at least 2 different types of organisms, and that's because, as we'll show you, the nutrient solution that we are using to feed those cells is derived, there's a part of it which is called fetal calcium, which derives from cows, and that's what feed the cells. Many human cells are actually being grown on a layer of what's referred to as feeder cells, and they are mostly mice. So often tissue culture experiments would constitute at least 3 different origins of organisms growing together to form a new form of life, if you like. So that's one thing. The second thing is that cells can be kept in suspended animation, so you can cryopreserve cells for quite a long duration of time, and you can reanimate them quite easily afterwards. And so that's, for example, what they've done with Dolly as well. The actual cells that were used to clone Dolly were frozen cells, and so you can hold the time, if you like. You can hold the gender and species, and you can mix and match in quite a lot of different ways that we wouldn't be able to do with any other type of engagement with life. So I suppose we should start. Adam?
Get a volunteer from the audience to hold up. You can use the rope even if you want. Now we have an anatomy table. Okay.
What we'll need is basically people starting to hack into that, and we will need a clear piece of bone, unopened, so we can put it inside, find a nice piece of muscle and cut it, and then we'll isolate the cells inside.
So they need some tools.
We have the tools.
Yeah. Okay, a pressure cooker will sterilize equipment. It's I don't know about kilos per square inch, but it's 25 pounds per square inch, 20 minutes at that pressure at how many degrees?
121.
121 Celsius. Yeah. And you have yourself sterile stuff. So we took knives and put them in gauze and then wrapped them in tin foil and then put them in here and pressure cooked them. You don't know what you'll find. Each one is different. Just put the knife back down, or whatever you have. In this case, it is a tweezer.
We're missing some person here. Stations have to be filled at all times. So Adam is now sterilizing the outside by putting ethanol. Being very careful in rubbing the right places.
Yeah. Ethanol penis.
Now,
ethanol penis. You want me to say it 3 times?
If you use 100% alcohol or 99% alcohol, what it does, it creates a layer that seals the living or the organic material, so the inside is still intact.
Okay.
Now, Revital, can I get the knife?
The idea is to try to get primary tissue from this body.
We need people to come and help us clear up some bones from here. So you have to cut through the flesh and start to clean out the bone.
Yeah.
Sometimes I'd like to be the cameraman.
We'll work with the ethanol later.
Yeah, good.
If you can just thank Mason for this amazing piece.
Yeah, Mason put together the box.
These are by far not sterilized, eh?
Yes.
Are there really living cells within the testicles, you think? Or?
Adam said that they got it this morning and that the animal was butchered quite, recently.
I'm volunteering to work with one of those testicles. If you could cut one off for me.
And you can work with it?
Yes. But you have the knife.
Hey, picture, yes.
You want to give me this?
No, too late.
Thank you.
Is there one saw you're not using that little one?
No.
Okay, let me
You can cut me.
rinse that.
Do you want to get it's such a funny thing. Do you want to test it?
The knife as well. Razor.
Jane?
May I use your knife? Hello.
Okay.
right?
May I use your
For
marker?
you.
Okay, that's maybe better.
Now he's satisfied. And I don't go to the meat section in supermarket. I want to go to
Very appropriate place to do so. What are we looking for, Adam?
Cells.
I know, but which cells do you want, Adam?
Did you cut it in half? So can you open it?
Do you want bone?
No, the bone we get from the female.
So what do you? So
Here I want muscle, so I want you to get, if you can get like a
but deep enough, right?
Yeah, very
so
deep.
these kind of next to the bone.
I think there's more like this.
This one, right? Yeah. It's basically contains everything the cells need to grow. So all of the amino acids, all of the minerals and vitamins they need, glucose for energy, and the color of the media is actually a pH indicator. This is kind of the basic of the tissue culture nutrients. To that you add, as I mentioned, fetal calf serum. So you usually add something like 10% fetal calf serum. Fetal calf serum is basically the blood plasma of a calf. They get the calf, even before it's born, they cut the cow open, they stick a needle into the calf's heart, suck out its blood while it's still alive, and then isolate the blood plasma, the serum. It's a problem in science because each one of them is different. So it's hard to maintain a consistency of research if you have something which is variable as that. But very few cells seems to be able to grow well without that. Then we also, just to be safe, we add penicillin, streptomycin, basically antibiotics. So if there's a small contamination, you hope that the antibiotics would treat that. We're going to use something which is called trypsin. Trypsin is basically an enzyme, a digestive enzyme that is being used to break down the glue that holds cells together.
Okay, and it fills up. To release it, like that. This is going to be kind of fun at home. Microliters is 1 milliliter. How am I doing? I'm not very good at this math stuff. So these are the pipette tips for these. These are also sterilized. There's this second lever, which is creation of biohazardous waste. So in other words, if you're pipetting, you want to pick up one of these, dip it into something, press down, suck up, move it into something else, press down and push all the way through, and then pop it in there and never use it again. Here's pipettes, and there's water here. Let's see if we can clear a little space for people. Is opening something like this with one hand, and then pipetting with the other hand, and then closing it. Now, if you can do that, then scientists think that you know what you're doing. You would pour in your potion, right? So pouring in a potion, it could be anything. One of the tricks of science is this. If you measure things well, maybe dilute them 10 times, 10 times, 10 times, and run an experiment, it can be anything because you're doing it in the right format. You're measuring well and it's sterile, right? In other words, if you have a control, and anything else, you can show an effect. If you have a sterile jar, which this is not at this point, you can push this through, and then that's now a clean potion. Right?
This does make me feel a bit sick. What?
Totally disgusting.
This is 1, 2, 3, 4. So you can do 6 and then 6.5, and you'll have 12.5. The hard part with the gloves is opening these. It's actually like that, and then you can also pull down and. Yeah, but I mean. Actually, the
Side of the actual stand.
if you're frustrated with getting a sample of the penis, you can move to a different part. It's not necessary that you be the penis guy. Laura, do you have any advice to your it's sort of tag team penis dissection? Do you have any advice? Maybe.
Yeah, I was working on the inside to get.
If you want,
No,
but would you like a sliver from the inside?
Yeah, sliver from the inside.
If you can get a sliver for Laura.
It's the right way around because I used another type.
Yeah, that is the right way around. I know it's open on the end. It's okay. Now, I'm going to watch. I know it's hard right now, huh? Very good. Yeah. Okay. Yeah, perfect.
So, how am I supposed to be efficient?
Well,
Because I find myself like,
I have to say that you have, first of all, you have the daring do. You're doing it. And you've gotten the bone marrow out, right?
yeah,
And the bone marrow is right there in your plate.
yeah,
That's more than enough for the entire class.
okay.
Okay, so you're being quite efficient.
Okay, but I don't know, I thought, I don't know, I think somebody told me I had to just, I don't know.
No,
Something
your
out
passion,
inside.
your passion has not
Okay.
gone ignored. Okay.
I can't hear. Sorry, man. We got like a thing in the middle. You want this? This in here.
We need 20 milliliters.
How much did you say?
20 mil in each of the jars.
In these.
Yeah. And that's to put the HeLa cells in. Oh, it looks better here. All right. We could use a little more light. Let me see. Just in case. If anyone wants to try this out, it's fairly basic. You can look through. So this is. We have how many? Who worked on the dissection of the penis together? Laura. I don't know. Oron, do you know anyone who's done tissue culture of, say, the kind of phallic muscle?
Muscle was not, but actually it was a guy. And you kind of shock them into life again, and then you keep it. And it takes them a while. Usually cells take about a week to recover from the trauma, from the shock of being frozen and defrosted. But some cells, in particular the HeLa cells, tend to be really fast. So what you do usually when you have primary cells, you do what we've done, you isolate the cells, you go through the whole process of trypsinizing them until you have individual cells. You put them on through those dropping degrees of antibiotic solutions until, so that's that process. The real process of isolating cells would take, usually I start in the morning, I finish only in the evening. But then once you're ready and you put the cells in the dish, you put it in the incubator, and some scientists actually pray to the god of tissue culture, and you don't look at them for about 3 days. You don't touch them, you don't look at them, you just hope. And then you start to check, and usually it takes anywhere between 3 days and a week, again, for them to recover from the trauma and start to grow. So don't get spillage. And that's for adherent cells, so that's for cells that stick, and they would with the primary cells, and then with the HeLa, we would put in some cells, put in the media, and then incubate it.
So I'll try to give a demo. I haven't played in this box very much myself. So this is not to protect us from what's inside the box, but to protect what's inside the box from us. Right? Use a meat tool to open your sterile tissue culture flasks, right?
You'd see a pallet at the bottom of your tube, which would be very, very small. So this is
that
in
a
a sense
huge amount?
way too much. And it's not chopped enough, but that's okay.
Yeah. Okay, so
But, you know, if everything was done right, there's still a good chance that out of that we would start to see cells migrating and growing out onto the dish in about 5 days.
yeah, not all tissue culture. There's other things that people use these flasks for, like organ culture, or like what we were talking about, explant culture, which just says more or less. Instead of just trying to make a cell line, you're trying to grow a piece of a kidney.
This one was actually, you know, what we really need to do is dip it with ethanol and then bring it in and then open it, and knowing that the inside would be sterile.
Where do I?
So where's the cleaners of the bone? There are
You need a...
rolls.
Oron, really quickly, will you open this and take this penis sample for the incubator?
I'll be proud. Thank you.
Oh, we got muscle coming down. What's the go on? You've got the bone marrow, I take it. It looks like it's moving.
But it's because of the liquid, maybe.
Yeah, I think so. Is this penis?
Layers of makeup become sexy.
Oh, it's not unusual. I had a friend who did a fibroblast culture, which is scar tissue culture, and his whole experiment was to run a sterile needle down the middle of the culture. It's just a monolayer of scar tissue on one of these dishes. Just ran a needle down the middle and then filmed them coming back in across the river of the needle scratch, right? They're scar tissue. That's what they do. They reach across barriers and then relink and knit things back together. Ron, do you want to do the honors? Please. Yeah. Okay. I will. Oh, stop it. All the cameras are moving in. Yeah? Yeah, that looks good. You can sort of tilt it up a little, I don't know, so it doesn't get in there.
Cross border control in the airport. Just put your belly in.
Yeah, act natural.
That's my third nipple.
I'll tell you a little story, because sometimes sharing is caring. The first time I did this lab on my own, teaching in New York, we isolated bone marrow in a homemade hood, and I asked my students to not just tape it to their body, but to write graffiti on the flasks. Not just write like this is penis culture and the date. It's okay to write the date and what cell it is once you isolate it. You work hard, you might as well. I just didn't bring 20 magic markers like usual. But I taped one on myself, and within 20 minutes of taping one on my body, I got a call from my father, who was stuck on the US-Mexican border with a balloon in his heart. And he said, "Get me the hell out of here." And I went online and I found like some doctor who does an open heart surgery called the octopus, where they leave your heart beating and they do one at a time, an octuple bypass with this crazy device that holds your heart up, and they do one at a time without stopping your heart, and shove it back in, and they literally sew you up with metal, like a wire. Your sternum, they have to saw it, they wire it back shut. He was walking the next day. He's still walking. What's that? I was like, It was 20 minutes after I put it on, I got a call. I was like, there is no way I'm taking this off my body till my dad is walking around, right? Till he's not dead. And he's still kicking, so just be aware that it might be a potent thing that you do. These, maybe you want to talk about it, these semi-beings, these partial lives are still alive. Right now, in here, there is a bit of life, right? And Oron speaks on this quite well, but I mean, I'm just saying, like, for me, keeping them warm is a relational thing. Talking to them is a relational thing.
So trying to treat them as entities which are more than what they are, which is really a bunch of cells lying in some liquid. And some people tend to feel that if they are good to them, the cells will be good back. So I need about 5 meals in each room. Standardize, so they were able, the way, in order to standardize them, they isolate one individual cell out of this cancer, which was the starter cell for all of the HeLa cells we have now. Started from an individual cell that was, in a sense, cloned and made copies. In the 1970s, a doctor who was working at Johns Hopkins Hospital, who was the place where those cells originally were isolated, had dinner with someone that her surname was Lux. And he found out that she was the niece of Henrietta. Now at the time, they were not sure if they are working with the original cells. So they wanted to know if the cells that are still known as the HeLa cells are the original cells that came from Henrietta Lux. And this guy got the details of the niece and invited her and her family to do some tests. They originally thought that the tests were to do with the cervical cancer. But basically, there were DNA tests to see if the DNA in the HeLa cells are still similar to those of the Lux family. And then the story came out, and the family actually found out about it. And since the mid-80s, they are campaigning to get Henrietta recognized for her contribution to science, because the HeLa cells were used for the polio vaccine, they were used for other things. And obviously, the medical establishment is really up in arms against it, because for them it's just a material. They don't want to recognize the person behind the cells. The HeLa cells were also used in quite a few artistic projects already, being such a symbolic cell as well. And obviously telling us stories about getting cells from a marginalized person in society, an African American woman, without her consent, being used by mainly white male researchers for their publications and for their careers. When you work with those cells, and as I mentioned before, there's no difference between human and animal. Those cells, you wouldn't even know when. There's interesting stories about research that was supposedly done on human cells, only to find out a few years later that those were not human cells. If you're not doing a specific DNA test similar to what those doctors did to the Lux family, you won't know where the cell originated from unless you follow the lineage of the cells. But as even in a very clean laboratory situation, not the messiness that we have here, there's issues where people might label a dish differently, or cells would contaminate and cross-contaminate. Where's Adam?
I'm here.
Okay, waiting for you.
I've been here for a while.
So we need to open the HeLa cells.
We're going to break the seal. Break the seal.
Break the seal, exactly. So, who wants to be honest? Okay, they were frozen in April 2008, so they were not frozen for long. I worked with some cells that were frozen in the 60s, and I opened up the veil, and they were still alive, so it took them maybe 2 or 3 days to come back to life. Those were frozen only for 1 year, or a bit more, longer than a year. And you can see this, what they've done here was they sealed the glass on the top, so we'll have to break the tip there. Who wants to do it?
How many tons of these cells are there in the world?
Because it's one of the most popular cells, there's tons and tons. I was trying to calculate how many tons of cells are all around, and obviously you can't really get the right details, but it's in the hundreds, hundreds of thousands of tons of cells.
And they are so popular because they replicate so easy?
Yeah, and they were the first, so the way scientists operate, obviously, if they do a scientific experiment to try to keep everything the same beside one variable, so because everyone started with those cells, they're still being used as kind of the gold standard for basic and generic kind of tissue culture work. And also they're really, really hardy cells. So some scientists consider them to be weeds.
Weeds?
Weeds, because they're actually contaminating.
How can they contaminate the whole lab?
Apparently, there's some proof that the HeLa cells can survive even when the dish dries out, or if when you work, there's sometimes droplets that fall off, so they would just survive in the sterile hood, and then if you touch something and you touch your thing again.
And then they multiply?
Yes. Those are amazing cells, though. Yeah, that's the thing with cancer cells. Cancer cells, first of all, lost their self-destruct mechanism, so that's why they can divide forever. That's why they're immortal. So, which is weird, but those are considered to be immortal cells because they came from cancer. Yeah. So, who wants to have the honor of reviving HeLa, reanimating HeLa? Should be one of you. I've done it before. Jump inside. The thing is, it's better if you open the vial on top of the...
Yeah, but then you have to guide me through because I've been in and out.
So you can see, you'll have to snap the top there, and then pour it inside one of those dishes.
Try not to knock too many things over, like I do.
I think those cells are so far removed from the human body that I don't think we'll be able to tell.
Thank you. So if someone wants to do this one, we can pass it to them. Have a seat. Come on now. Yeah. So, Oron, is the idea to break it and then pour it in?
Yes.
And
try to break the tape of it.
Good.
And then pour the tape in the rest into the dish.
Your hands.
No, use the tweezer. You can try the tweezer.
Yee. Okay, can I pour it in here? Is that it?
I think you pour it in here. The idea is you use that to keep it from spilling on the wood.
Alright, I'm not sure if it's open yet.
You can try a test.
How many drops?
Yes.
Question is how many drops?
Half of it.
Or maybe 1/3 because we have 6 people.
Are you dripping cancer?
Do we have the marker?
That's it. Good we have the hood. Half of it or 1/3?
Take a towel and really break the hole after.
This is a very dirty way to get how we go today.
Adam, did you have?
Yeah?
Is there another pen?
Yeah, we're passing it around. Anybody need some? You're all set?
Yeah, so just put markers. It's better if you put them in the incubator as soon as possible. So. So you can see how different this is from the primary tissue isolation. So very clean and neat. So most scientists would never see and never even realize what's the original organism from which their cells were taken. You know, they might read mice, but it's so far removed from them. This is human. Those are fragments that were taken from human cancer. How apparent is that?
Apparent.
Apparently.
Where is she going now?
Incubator.
And pass over this way.
And how long will it take?
Hopefully by tonight, but after the lecture, we'll look down the microscope and see. What you'll see now, if you look down the microscope now, you see those round little things floating. But if we're successful, they'll settle down and will spread out. And we'll see the cells. Now, a very important part of lab practice is not just getting ready, but also cleaning up.
There's two more here, and then Lucas isn't. I have to label mine, so let Lucas pass those two.
This one's got cells in it as well? I don't know. There's no label.
No, that one doesn't have cells.
Okay, let's control then.
Yeah.
Right control.
If anybody would like to take a sample, there's even still time. You can hang out after and tape one to their body for the experience of being an incubator of something unknown. You're already doing it. How do I say? I think this is a good way to determine what tissue culture is for general people. I don't think there's anything but general people in the world. There are no real experts of everything because we haven't figured out what's going on. But for me, this is a way to help people speak about tissue culture, about what it is and what it isn't, about biology, what it is and what it isn't, and also be able to say, oh, I did that, and not be intimidated by someone who's saying what they know. And it's incontrovertible because they're from the world of knowledge and you're not. So I hope that you take with you some imparting of some sort of personal way of knowing this. I'm not sure what it is either, but I think that hands-on experience, even with gloves and containment, is an important facet to, how do I say, not just public understanding of science, but public conceptualizing of science, right? Because it's sort of not just up to who makes up new technology as to where and when our bodies get consumed by it, right? So I hope you take that with you. I don't know. We'll see. And I thank you. There we go. Really what we should do is now autoclave this in the pressure cooker, but we have them alive over there. I'm crawling in them. But that's a big amount.
Can they be infectious in some or another way? We should ask Oron.
Well, we talked about it actually quite a lot. That they say it can't, but that there are Tasmanian devils in Australia that are dying from an infectious cell line of cancer that they pass from scratching and biting each other.
it's nice if you, as a living organism, creep into the glove box.
Oh yeah. See if I can do this.
Replacing the HeLa cells that were in there just. Quite a bunch of living organisms in here. You're cleaning all the gloves?
I'm sterilizing these gloves before the performance. Make sure they're clean.
When will the cells that you carry on your body start to?
Oh, you mean these guys?
To grow?
Well, after 4 hours, they should attach to the side of the flask, and then sometime tomorrow, I should be able to look at them under the microscope and see if they've been happy here.
Yeah.
We'll see.
And you need to keep them on 37 degrees.
37 degrees. You should test to see if I'm getting fat enough. You know what I mean? Like Hansel and Gretel. Do you feel my finger?
No, stay in there for a little while more.
Literally, if you have one of these flasks, right? If you have the cells in the flask and they're on the bottom, they're crawling around on the bottom. But in 3 dimensions, it's just unusual 3 dimensions. Like you see them from above in a microscope, they look 2-dimensional, but if you make a film, they're moving. They're moving about, they have dimension. If you add something in there of a sculptural nature, like a glass bead or a glass sculpture, or something like fibrous, you know what I mean? Yeah. You can then, how do I say, they'll grow into it. They'll grow into that, you know? Ooh. I'm gonna open it up. There you go. Don't look. Oh, you naughty. I mean,
yes or a no?
I think actually there should be more scientists like me, not only that are willing to do casual cultural understanding, social interface work, but are willing to have fun at the same time, and also touch on touchy subjects that are, you know, bioethical subjects that don't necessarily please, say, the corporations. Not all scientists work for corporations, or please even the scientific status quo, but even to allow for scientists to be expressive beings too. You know, they're out there, so I'm like, I make a call for the scientists, because artists, there's plenty of expressive artists. Now, artists that understand what they're talking about, especially if they're critical of new technology, it sort of helps for them to know what they're talking about and to show that they know and that it's not that hard to figure out. I don't really just differentiate between lay people and experts, because nobody knows everything, and being an expert can be it can actually be a disability. In other words, you presuppose what you know, and actually what we know is much less than what we think we know. So regular people, my people, regular people, working people, just to, you know, baseline existence people are fucking hella smart. And so it doesn't hurt for there to be a large amount of people that understand the issues and the technologies to a degree where they can actually say, like, you know, it's not that big a deal. A bone with bone marrow just growing in it isn't, like, you know, gonna save the world, but also, you know, that they're remaking us one bone at a time, this sort of thing. It's interesting. Taste is just like, it seems to sound similar to it. Most people think aesthetics is what is beautiful. It's not, it's what you feel. So, like, if you feel repulsed, that's a feeling. If you feel smug, you know, kind of like, that's a feeling. If you feel like depressed, that's a feeling. And depending on not just the range of feelings that you're capable of, but also the range of affect, the range of emotion that you're capable of feeling, say, depression, or smugness, or joy, or beauty, that's the degree that you can appreciate an aesthetic. Taste implies that there's better or worse taste. Feeling.
How do you feel working in the context of Waag Society as a cultural organization doing things with creative technology?
Well, I mean, I think the major two things I would say is, one is, what is cultural and societal use? In other words, what is useful for culture and society, not for culture and society, but the people in the cultures and societies, because I still, even though I'm not into the cult of the individual, I believe in people, individual people, as being part of society, but not the society making them. Number two, on that note, what's useful to a society is not necessarily utilitarian. I think you guys do a lot of interface network stuff that's about creative, subjective interaction with disparate cultures, global feelings, and that to me is more important than just survival, because we have problems with sharing in the world, but one of the problems that leads to the sharing not happening is misunderstanding of the diversity of people. And so I'm sort of interested in how that can work. On the level of biotechnology, I mean, I think taking it to the streets like we've been doing, working with various different cultures, like if you guys are global, I'm gonna go around the world in this box, you know, let's go. But I think also the way that we're reaching a point where the future of human anatomy is going to be determined by either very few or very many, you know, and it's going to be determined in different ways in different cultures according to different aesthetics of feelings, whether it's joy or disgust or pride or pornography. So I'm kind of interested in analyzing that, and I think the Waag is the kind of place that does things like that, like seeing how people interact with these type of new technologies on a gut level. Yeah.
But I
Okay.
Hey, baby.
Can I have
Can you have a what?
a little drop of alcohol?
Ethanol?
Do you? Yes. That's perfect.
Okay.
I can add water to that?
Yeah.
Do you want your syringe back?
Do I want my what?
Your syringe back?
No, that's okay. Okay, somebody's got to help me out of here. Yeah. Well, I mean, first of all, as many scientists will agree in public and maybe disagree in private, scientists don't get to decide what their discoveries become. Right now we have an awful lot of corporate and government funding of science for discoveries that will be utilized in the traditional way, which is to increase productivity, increase weapons development, and perhaps sometimes increase human lifespan, for better or for worse, to keep people working harder longer, maybe dying faster when they can't work. What I'm mostly focused on is human re-engineering, right? So what some people call enhancement, I don't think of as enhancement. Getting smarter and more beautiful and taller and living forever, that's not what I would call enhancement. That's a particular aesthetic. All right, and not just better, as in good or great, but actually a particular traditional way of thinking about what's better or great. Biotechnology has a history of being involved with these type of things, and this history is being revived in the realm of genetic engineering mixed with say like family planning and breeding, right? So pre-implantation, genetic testing, strange things that are going to be offered at fertility clinics, technologies that have to do with reversing sterility. All of these sort of new technologies have the potential to also add genes at the same time, at that very moment when a new embryo is being formed. It's not a new idea, it's not paranoia. We're making new kinds of humans, and in the name of, the space race, or, new biological warfare resistant soldiers, people are willing to donate whole lives, whole beings, to experimental beings to see if we can get one that will make a better soldier, make a better astronaut, and eventually make a ton of money, right? Like, what if we make a human that's part spider? Not like Spider-Man, but with 8 legs and 8 eyes and hair and likes to eat their mate, this sort of thing, or spin a web around normal humans and suck them dry. That sounds monstrous, but it's actually, it could be a post-natural kind of naturalism compared to the idea of just bigger frontal lobes or bigger tits. This sort of standard, I'm not against populism, but this sort of standard dream, which may be being funded quite well, even as we speak. Evolution tries everything. And sometimes it sees what lives, and sometimes it sees what's willing to cooperate with the environment and other organisms, like altruism, mutualism exists. But nature doesn't have a taste, nature. So, do I think we know what we're doing with the human genome? Do I think that we understand the metabolism even of a bacteria? No. Is it genetic pollution? Probably. Will we get the results we're looking for? Sometimes. We'll have to kill a lot of embryos to get the one that looks just right. But will it be happy with our aesthetic imprinted upon it? I'm not sure. Will they, our kindred, will they thank us, for making them smarter just in the rational area? Not smarter in the hippocampus with a wider range of emotions. That's not funded. Frontal lobes, neocortex, that's funded. What are we doing? That's my question, actually. And something like this, right? So the transformation from the visceral to the sterile and contained, right? And we're showing people it's not that far from what you do at your dinner table to what science is doing. It's not that far for you to be able to fathom, right, what science is. Hands-on experience makes you implicated, right? Like this tissue sample here that's incubating on my body. I'm implicated in the actions of science. So in some ways, it's actually a trick, right? I mean, that people have the right to say no, but much like the Milgram effect, when allowed, you may find people that ordinarily you thought would say no, don't say no. They actually are interested. And that's kind of interesting to me. I always like the students that say no, that I say, well, you're our ethical advisor. You're on the outside. But to tell you the truth, being able to say I did that, and I know what it is to me, without someone from science saying, we're making a fact today, but instead saying, does anyone feel uncomfortable about this? Do you want to express that? For people to go home and dream, not that I did a scientific lab today, but that I did a lab that involved science that had to do with my own feeling about what I was doing.
Okay, fine, thank you.
Sound good?
Yes? Oh. One practical question.
Yeah.
We may only use this for broadcasting if you explicitly give us your permission. So could you please on camera...
All right, Zoot, my ethical advisor, if I give on camera my right for broadcast, is he allowed to make $1 million on me?